All right, I think we can get started. So, good afternoon. Thank you for joining us for the FHCC Connect Science seminar series. And as just reminders, please post your questions in a Q&A function of the Zoom, not in the chat. And also, as you saw, this seminar will be recorded and will be posted on the website in a couple of hours after the seminar, so you could check it back. And it's my pleasure to welcome today's speaker, Amy Brock, who is Associate Professor in the Department of Biomedical Engineering at the University of Texas, Austin. And she actually did all her training here up in Boston at Harvard and MIT. She was an undergrad at MIT and then completed her PhD in the BBS program at Harvard, and then also did her postdoctoral training at Boston Children's Hospital and the Weiss's Institute for Biological Engineering. And then she moved to UT Austin in 2013, where she has been since then. And she's been very productive and very innovative throughout her career. And most recently, what she will share with us is this very exciting technology that she developed that allows the tracking of populations of cells. The title of her talk says it what it will be. It's tracking population heterogeneity and emergence of chemoresistance with functionalized cell barcodes. It's a really cool system, and we're very happy to have Amy join us today. So thank you, Amy.
Thank you, Nelly, for that very kind introduction. We were just talking before the seminar began about how Nelly's career has really inspired me for many years. And so I'm so happy to be here and join you all to talk about our latest work. I'm going to hide the video. So if anyone has a urgent question to ask in the middle, please, go ahead and alert me somehow if there's any issues with the technology. So, my lab at UT Austin is really broadly interested in understanding the fundamental functional roles of heterogeneity in mammalian cell populations, and we're especially interested in the role in therapeutic response and the rise of resistance in cancer cells. And I think for this audience, I don't need to convince you too much that variation among individual cells within a tumor is interesting and has important implications for disease progression and for treatment. And in addition to looking across different patient tumors at intertumor variation. So I'll go past that. So really, to understand the contributions of heterogeneous cell states, what we'd really like to be able to do is to track individual cells through the long-term emergence of resistance. And in some past work, which is going back to when I was a postdoc, published in 2013, we were able to do this. This was with Sui Huang at Boston Children's, now at the Institute for Systems Biology. We were able to do something like that using both wet lab and mathematical modeling approaches because we had a fluorescent substrate for a resistance marker. So in this study, we were looking at the expression of the MDR1 protein or P-glycoprotein in leukemia cells. And we were able to track that expression via a dye-based reporter or via antibody labeling, right? But when I thought about how to broaden these studies, an aspect of this is very challenging is that these require some a priori knowledge of what is the resistance mechanism that's at work here. And in a real world setting, that's often not well defined. And as well, that is likely to change over time with disease progression, with treatment. So I really felt that we needed an unbiased kind of marker system that we could use despite having no previous knowledge of what was the underlying resistance mechanism that might be at work in the population. And so after, thinking about this for a while in my own lab, we decided that labeling cells with random nucleic acid barcodes would be a way to accomplish this. And this particular design of a barcoding platform was the work of Aziz Al-Khafaji, who's a PhD graduate of my lab. And we first published this technology in 2018. And the way that it works is that every cell in the founder population becomes labeled using a viral vector for delivery, becomes labeled with a unique random 20 nucleotide sequence that also encodes a guide RNA. So the nucleotide label is integrated into the cell genome. It's inherited to all of the daughter cells. And in addition, the cell expresses that unique guide RNA sequence. And the 20 nucleotide random bit of the sequence is the CRISPR recognition site of that guide RNA. And the backbone that you see here of the vector is from the crop-seq design developed by Christoph Bach's group in Vienna. So with cells that are tagged this way, we can perform several kind of unique types of studies. And those are shown on the right here. So first, from the cell population, we can measure clonal composition by performing targeted sequencing of the genomic ensemble of barcodes. So starting with the regions just flanking the barcodes. And so in this kind of data, the read counts will correspond to the abundance of that particular clone in the population. Second, because these barcodes are express sequences, they can be read in single cell RNA-seq workflows to yield lineage resolved or clonally resolved single cell transcriptomes. And I'll use those terms somewhat interchangeably, lineage and clone, throughout the talk. And finally, at the bottom here, we can go a little bit farther with this design and use the guide RNA barcode not only as a static label for quantitation of clonal dynamics, but also here as a kind of actuator. So we can use it to pinpoint specific clones within the overall heterogeneous population and use that barcode to drive expression of a reporter gene. And that aspect is pretty unique to our system. And I'm going to come back to that piece and show you how it works at the later part of the talk. So a feature of all of these kinds of barcoding technologies that we really like is the ability to compare the behaviors of clones across many replicates, right? To truly have the power of replicates. And if we create replicate samples from the same starting population, they should each contain the same collection of barcodes, if we are careful and perform our math correctly. And so that means in parallel treatments, we can ask questions about the origins of the clones that went out. So for example, in this schematic where cells are treated with a selective pressure, let's say it's a drug treatment, we can look for in the data two potential outcomes, right? So all of the replicates might display very different barcode compositions, which would be assigned to us that there's some kind of de novo process at work. It could be a mutation or it could be epigenetic change, but some kind of alteration that's determining the survivor cells, which means that many different cells in the starting population have the potential to survive the treatment, right? Or conversely, we might see that the very same clones, consistently survive across the replicates, which would be an indication that there's some pre-existing resistant cells in the starting population that are selected for and end up being the survivors across many different replicates. So I really love this approach because it lets you essentially ask these kind of big questions, like Gould did for macroscopic evolution, where he asked if you could replay the tape of life on Earth, what would it look like each time? Would it be the same? Would it be different? And I think here the power of these barcodes is in part the ability to replay the tumor's evolutionary trajectory and ask, whether we get the same results. Do we see the same initiating cells contributing to the disease? Do the same ones survive therapy and contribute to relapse? So DNA barcoding is really perfect for this kind of quantitative assessment of clonal fates. So we had this system up and working, and some of the technology was published in 2018. And we decided to deploy it in a first study in a collaboration with Kathy Wu, who is there with you at Dana Farber. This was a collaboration with Kathy and Kathy Gutierrez, who was an MD PhD student in her lab, really drove this study. So we used this system to look at the clonal responses of CLL cells to cytotoxic chemotherapy. So as you know, CLL is a B-cell malignancy. The relapse rates are about 20% in 2 years, the first 2 years. And so we decided to focus on this relapse type setting and use the barcoding approach to learn about the origin of those cells that survive chemotherapy in vitro, and in a clinical setting, perhaps maybe contribute to disease relapse. So Aziz from my lab worked together with Kathy Gutierrez in the Wu lab, and this was a really close and dynamic collaboration. We sent reagents, but also people back and forth between the groups. And so please, whenever I use we in the rest of this section of the talk, it refers to the two groups really working together. And this was really driven by the graduate students whose work really made this happen. So Kathy and Aziz worked together to establish barcoded CLL cells from the HG3 cell line, which is one that harbors a common alteration, deletion in 13q. And then they split that barcode labeled population into 8 different replicate cultures, such that the same barcodes were represented across each, and treated each replicate with a combination of fludarabine and mefosfamide. So that's the active in vitro analog of cyclophosphamide at a high dose, at a high LD95 dose for 72 hours, and then washed out the drug and allowed the cells to regrow. So, and this takes about 4 weeks for those populations to grow back. And then we were able to use the quantitation of barcodes to ask about how did the composition look across these replicates? Could that tell us about the origin of the cells that are in the surviving regrown population? And so when we quantify barcode composition by sequencing the ensemble of barcodes, so first what you see in the left is that there's a massive reduction in the abundance of unique clones. So many clones are sensitive to the treatment, so that makes good sense. And then when you look at the heat map in the second plot here, we found that most survivor clones, so every row in this plot is the abundance of one barcode or one clone. We found that most survivor clones appear in multiple replicates. Another way to look at that data is as stacked box plots. So about 30% of the clones are shared in common across all 8 replicates. They survive in every replicate, and about 90% are shared with at least one other replicate. And in addition, the last bar plot shows you as a percentage of total population, the shared clones comprise the majority of the post-treatment cell populations as well. So this was some good evidence pointing towards a pre-existing resistant state that's already present in the pre-treatment population that contributes to the survival of those cells. And we next wanted to characterize gene expression profiles that underlie these survivor cell populations. And So we can do this using the 10x Genomics single-cell RNA-seq platform because the barcode sequences are integrated and also expressed. We can read them out along with the other transcripts in cells. And so this analysis was the work of Eric Brenner, who's a graduate student in my lab getting ready to defend in a couple of weeks. And so we performed single-cell RNA-seq, lineage-resolved single-cell RNA-seq of 2 of those post-treatment replicate samples, and those are shown here in purple and blue at the post-treatment time point 1. And then as well, the pre-treatment sample at time 0, which is shown in green. This data represents about 9,000 different cells, and you can see they group into 8 transcriptionally defined clusters. The pretreatment cells, which are again the green, group into 2 distinct clusters, and the post-treatment survivor cells fall into 5 somewhat tightly or related 5 somewhat overlapping clusters. And remember, for each cell, we're able to measure its transcriptome, but also we know its clonal family from the barcode identifier. So on the rightmost UMAP here, you can see labeled the 10 most abundant survivor clones. And just showing you 10, because after that, we start to run out of colors that we can distinguish, but there are 1,050 different clones labeled in this data. So most of these gray dots, for those cells, we also have a barcode ID that's associated with those as well. We have barcode data for about 91% of the cells for which we also have RNA-seq transcriptomes. So now with this type of data, which is kind of a new level of resolution to look at single cells, we can use the barcode identifiers to track the trajectories of clones and follow the populations, right, from pre-treatment to post-treatment. And when we do that, what we learn is that all of these, I hope you can see the pointer, but all of these abundant survivor clones in this large red grouping that comprises 95% of the survivor clones, the post-treatment population, they all originate, we can trace back those clones to cluster 5 in the pre-treatment population. Cluster 5 is a minority of the pre-treatment population, but all of these abundant survivors come from that population. While all of the cells that end up in cluster 6 in the post-treatment population, they're about 5% of the post-treatment population. They all could be traced back to have origins in cluster 2, which is about 80% of the pre-treatment population. And there's almost no overlap. So we're seeing here 2 discrete gene expression states in the pre-treatment population, one that's composed of a unique subset of clones with high survivorship potential. The other is composed of a subset of clones with low survivorship potential. And their transcriptomic responses to chemotherapy are very much preset before exposure to the drug and linked to their clonal identity. And almost no cells deviate from these 2 different trajectories. So we performed marker gene analysis, which is shown in the heat map on the left, and saw that the high survivorship subpopulation in pre-treatment has upregulated Wnt signaling, markers of inflammation and proliferation like CXCR4, JAK1, and the low survivorship subpopulation was marked by upregulation of adhesion markers, CD18, which is ITGB2, and FGF receptor, among others. As an example, these heat maps shows the distinct profiles of CXCR4 and CD18, which we're going to use in a couple of future slides as well. From the heat map, one other point that I think you can see, even though I know it may come out small on your screen that's at home, are that both the high and the low survivorship profiles are largely maintained following treatment, so they're relatively stable phenotypes. And with our collaborators in the Wu lab, and thanks to many at Dana Farber, we were able to perform a comparison with patient samples of matched transcriptomes that were collected before and after relapse to fludarabine-based chemotherapy in a cohort of 23 CLL patients. And what you can see here is that we confirmed a marked enrichment of the high survivorship, but not the low survivorship signature at relapse, and specifically of CXCR4 and Notch signaling that were significantly enriched at relapse in those patient samples. So to learn more about the different responses of the high and low survivor populations, and I'm going to quickly summarize a lot of work that Kathy Gutierrez did to look at this. We examined an earlier time point as well in the recovery of the cells from drug treatment. So the 0 time point is pre-treatment, and I've been talking to you about this post-treatment time point about 4 weeks after the cells have fully repopulated the initial population, and now we have an initial time, a new time point partway through that recovery. And so we looked at earlier time points in the regrowth from drug treatment, and we found, looking across all of these, that both the high survivorship subpopulation and the low survivorship subpopulation had markers of apoptotic stress responses. But the low survivorship population was more primed for apoptosis even in the pre-treatment time point. And GSEA analysis showed that the high survivorship clones relied on oxidative stress and DNA repair pathways that were upregulated, and many genes in those DNA damage response pathways, DDB2, FDX, and NJ1, these were already expressed at elevated levels in the high survivorship versus the low survivorship subpopulations, even in the pre-treatment time point. So indicating that there could be some priming of those repair pathways in that subpopulation, even before the cells saw the drug treatment. So another aspect of this that was really intriguing to us is that these two subpopulations, they coexist, and their proportions remain stable over time in culture as the cells are propagated. And we were really curious to learn about that process a little bit more, to learn whether these subpopulations interact in some way to maintain their ratios. So we used the barcodes to measure first whether there were any differences in growth rates in the parental population. So this is another real convenience of having barcodes, right? So we have now the ability to categorize the barcodes based on phenotype, high survivorship, low survivorship. Now we can look back at growth data collected as the cells are maintained and cultured normally, and assign those categories to the different barcode populations and compare them. So we looked back at the parental population during the expansion, just through normal growth, to see if there were any differences. And there weren't. That's what's shown in the histogram here on the bottom, and this comparison. We couldn't find any differences in that context. But if we used the cell surface markers that we identified through the single cell RNA-seq analysis, and we sorted out the CXCR4 positive, CXCR4 high subpopulation, which was a high survivorship subpopulation. It's going to be red throughout. And the CD18 high subpopulation, which is the blue, the low survivorship subpopulation. And then we culture them separately. Then we start to see some really interesting differences. So in this context, the high survivorship subpopulation exhibits a significantly faster growth rate compared with the low survivorship subpopulation or the bulk population. We then we took these two subpopulations and labeled them with fluorophores and set up co-cultures, right? So we co-cultured the two subpopulations in a range of starting proportions. So we varied the initial proportion of each population, and then we monitored those over 14 days. And what we found was that in cultures where they were comprised of only 10 or 20% high survivorship clones, the CXCR4 high clones, those populations, which correspond to, by the way, to the parental cell line, right? So to the HG3s, off-the-shelf HG3s were in this range of about 20% high survivorship clones. Then they displayed a stable population composition over time, the same as we observed in the bulk population. But if we got to proportions where the initial abundance of high survivorship clones was beyond 30%, then their frequency continued to rise over time, but not at the rate of the high survivorship clones culture in isolation. So they never sped up as much as the high survivorship clones purely isolated by those 100% in 100% culture. So those findings, really suggest that the low survivorship clones are imposing some anti-proliferative effects on the high survivorship subpopulation, right? We tried, but were not able to confirm a biological mechanism to explain that interaction, but we did identify some candidate ligand receptor pairs predicted from the single cell data using the cell phone DB tool. And some of these, especially LGAL-S9, SORL1, have also been noted in patient data. So this may be a place for future study to try to pinpoint a biological mechanism there. Great. So I did say that we have this other facet of the barcoding system and this functionality that we haven't yet talked about that allows us to evaluate subpopulation heterogeneity at a little bit greater resolution. So now that we know through these other studies that some clones are of particular interest to us based on their fate or their high abundance under selective pressure, we have the potential to go physically isolate those specific clones from the bulk population, which contains many thousands of unique clones, by going back to archived samples frozen at different time points or continued in culture if that's feasible, using those guide RNA sequences to drive transcriptional activation. So this feature is kind of unique. So we're pretty proud of it. So it's a very simple system. It's accomplished by delivering through nucleofection or transfection of the whole cell population this gene circuit. It consists of a Cas9 transcriptional activator, which is the dCas9VPR transcriptional activator, plus a reporter plasmid, where we have placed the barcode that corresponds to our target clone, and that's shown in yellow here, upstream of a minimal promoter and a reporter, a fluorescent reporter sequence, so that only cells that are expressing the matching guide RNA will be able to recruit Cas9 to the promoter, drive reporter expression, and those can be isolated by FACS for future live cell assays or growth or other types of characterization studies. And so, sometimes when I'm on study section or so, I hear the caveat that, a single cell omics is limited because it's a destructive measurement. And that's certainly true. We can't make parallel measurements on the exact same cell here in this context either. But what we do have the ability now to do is go back to a sister cell or a grandparent cell or however you'd like to call it, and another cell in the same clonal family, and perform complementary types of studies. So in the CLL cells now, to look at subpopulation heterogeneity at a little bit greater resolution, we invoke this ability to perform clonal isolation. We isolated out 2 clones that were present in the high survivorship subpopulation. So these are survivor cell resistant clones. And when we isolated those clones off the sorter, we did another step where we seeded them into individual wells so we could get subclones as well as the major clone. And so given that the clonal families have displayed so far these consistent phenotypic responses, we looked at differences in SNVs by whole genome sequencing of those lineages. And this was done in collaboration with the Broad Institute. We looked at differences in SNVs using the low survivorship subpopulation as the reference here. And what we found was that between these 2 isolated lineages, about 40% of mutations were shared, indicating a likely common ancestor, but most mutations that were observed were unique to one lineage or the other. Following exposure to therapy in that post-treatment sample, there was increased diversification of the isolated lineages, which makes good sense. There was about a 2-fold increase in SNVs in both. We identified on the leftmost table here, we identified 33 mutations that were predicted to have a functional impact, given that there were missense mutations and based on polyphen scores. And one of those, TRIM-L2, is a P53 regulator that's associated, has been identified as associated with apoptotic responses to oxidative stress, and has been identified as upregulated in other cancers, particularly oral cancers. This one was elevated in our cell fractions that survived therapy as well. From those, each of those 2 major clones, we also isolated and sequenced 3 subclones. So among these subclones, more than 73% of SNVs were shared among the 3 subclones. About 27% were unique, and that means probably acquired during their expansion from their common barcoded ancestor, which was fairly recent in the course of this experiment. And again, following treatment, if we compare with the numbers with the post-treatment, the time point 1 sample, there's about a 2-fold increase in identified new unique SNVs there as well. So the high survivorship subpopulation, I think overall what this whole genome sequencing tells us is made up of numerous genetically distinct lineages. These are just 2 of them, right? We could have looked at more. The unique SNV signatures of the subclones here are maintained over the course of expansion of the cells and the treatment of those cells Although the subclones are also constantly diversifying, and that's happening both in the absence and the presence of these strong environmental pressures. So I think that was an interesting piece of data that came from this analysis. Given this high degree of genetic variation among the clones, we also wanted to look at their transcriptomic heterogeneity. So we performed single-cell RNA-seq on these 6 isolated subclones from the pre and the post-treatment, each one. So we hashed and ran those 12 samples together. So we had about 500 cells per subclone that we analyzed in this data set. We found, if you look to the UMAPs first, that the cells largely segregated based on their treatment status and based on their lineage identity, recapitulating in this dendrogram their clonal relationships. Then we asked whether it was possible to take this new knowledge and classify an unknown cell by lineage identity, using this data. And so here, Eric Brenner in my lab used supervised learning approaches, and he generated a multivariate classifier to learn the transcriptomic signatures of these 6 subclones within each time point. And he found that this model was highly accurate. You can see the scores of the ridge classifier that he developed compared to a dummy classifier. The weighted precision scores were higher than 0.97 for both the pre and the post-treatment data on those subclones. So there is robust transcriptomic variation within these different subpopulations. Given that encouraging result, Eric then asked whether these characteristic clonal transcriptomic signatures were stable over treatment. And so this kind of inspired him to look back to the 10 most abundant survivor lineages from that initial single-cell RNA-seq data set that I showed you, and try to classify those lineages by their lineage identities. So this was now a 10-class model, a little bit more complicated situation, and ended up with weighted precision scores of 0.73 in the pre-treatment data and 0.83 in the post-treatment data. The pre-treatment data contains, in the pre-treatment data, because these lineages are more rare in that data set and are make up a greater proportion in the post-treatment data, I think it's a much harder, much more challenging problem in the pre-treatment data. But nevertheless, again, weighted precision score of 0.73, pretty respectable, supporting the idea that the lineages have some stable characteristic transcriptomic signatures that get conserved over the course of treatment. By ranking genes by their weighted coefficients from the trained model, and here we're visualizing the expression of the top 20 genes for each lineage across the 2 time points. We observed that most lineage-specific signatures, they're really comprised of a constellation of gene expression values. So there's not any single gene that we could point to as characteristic of a specific lineage's transcriptomic signature here. So again, altogether, I think these observations give us a way to link the accumulation of genetic alterations to transcriptomic variation and the development of characteristic subclonal signatures that get maintained over expansion and treatment. In this section, and this work was published a couple of months ago in Nature Cancer. So to just kind of remind you what we found in this study, we used the express barcodes to elucidate trajectories of cells that were responding to chemotherapy. In HT3 cells, we identified 2 discrete subpopulations with differential drug sensitivity and different transcriptional programs. And using this approach, we can start to quantify a balance between genomic diversification and transcriptomic stability, and hopefully have new tools to systematically dissect clonal dynamics in a cell population. I'm going to switch to talk about some newer data, but that uses a very similar experimental scheme. So let me tell you about sort of what we've got in the works right now. So one thing that we've been doing, looks very, very similar setup, is to, so my lab has done a lot of modeling using various breast cancer cell populations, and we recently were awarded a U1 in the Cancer Systems Biology Consortium to look at triple negative breast cancer cell models and patient-derived cells, and look for subpopulation heterogeneity in that context. So we're starting to bring these tools to bear on that problem. And this is some of our first results along those lines. So here you can see a similar experimental setup using the MDA MV231 cell line, which has been barcoded and split into multiple replicates and treated with the chemotherapeutic agent doxorubicin, again at a high dose for about 48 hours, and then the drug is washed out and the cell is allowed to repopulate, to regrow. And I'll show you, so first of all, you can see again, the number of unique barcodes decreased significantly. We started with fewer barcodes. This time, I think we're learning along the way that we don't need necessarily to have tens of thousands of labels to cover the diversity of cell population. So we're learning a lot about that as well. You can see that across the replicate populations, there is about 10 to 20% of barcodes that are shared across replicates in this study. So it's there's some signature of a pre-existing resistant population, but there's a little more variation than there was in the example that I showed you of the HG3 cells. And let me show you some UMAPs that relate to how these cells are behaving at the single cell level. So I'll show you here 4 samples, a pre-treatment population of barcoded 231 cells, 2, which is shown in purple, 2 of those replicates that have been treated and allowed to regrow. So we see the survivor cells that are shown in blue and green. And then an additional time point that's collected about 30 hours into the treatment window. So the cells are certainly responding to doxorubicin, but there is not yet significant change in viability. Again, we have barcode identifiers for about 91% of the cells, for which we also have transcriptomic data. And so I labeled about the 12 most abundant lineages in this data. So let's talk a little bit about what we've learned so far from this. The first thing that was at first a little bit of a surprise to me, but has been reported by others, in particular Adam's lab at MD Anderson, is that the 231 cell culture, which is sort of a workhorse model for many of us, as an in vitro TNBC cell line, breaks down into these 3 gene expression states. And it turns out in this study, the clonal composition and the fates of the cells in these different clusters is different. And so if we track the clonal dynamics and follow these clones from pre-treatment to post-treatment using the barcodes, we can see how each of these three populations ends up having a different trajectory. So the cells that start out in this highlighted subcluster in the pre-treatment population, at 30 hours into treatment, they show many indications of doxorubicin response, and then they all die off. So those never appear in any resistant populations. None of the clones that originate in this cluster go on to survive. The cells the clones that begin in this pre-treatment population have sort of their own unique little trajectory. And it's dominated by one particular clone, which is shown in pink, but there are a number of others that are less abundant, but still present in this cluster. So if you squint, you can see a lot of gray dots here and here as well. And they have a different kind of initial response to treatment. They end up comprising a minority of the survivor cells, but some of them do manage to survive for sure. Finally, and then if we go and look at the signatures of these survivor cells, we learn that they display multiple markers of p53 response. The GSCA analysis also picked up SPDEF. ESAM is a cell surface marker that has proven to be useful in purifying this subpopulation out, and as well some signatures in cell adhesion alterations. Now, all of the other cells, so I said this is a minority of the survivor cell population, and all of the other survivor cells, which are in these large clusters to the right, they all originate in this central cluster from the pre-treatment population. So they have trajectories that look like this. They all move upon initial acute treatment of doxorubicin to this cell state, and then they end up in one or another of these survivor cell states or resistant cell states in the long-term regrowth. And so every clone here can be traced back along this particular trajectory, but being in this state, this cluster pre-treatment, is not sufficient to confer survival. So there are still the majority of clones that began here die off upon treatment. So it's necessary in this case, but not sufficient to start out in this state to survive this particular selective pressure. There are these two distinct resistant cell states that we observe, and it's possible as we continue to collect more data that there'll be others. When we compare these in terms of gene expression signatures, the one that's highlighted in bold here, that's dominated by red, but there's also green and orange and other lineages, clones present in this particular cluster, show an upregulation of RAS and MAP kinase signaling and Rho GTPase signaling. And I know these labels are small, and I apologize for that, but this is comparing this signature in this resistant cluster versus the 30-hour cluster, the pre-treatment cluster, or the second resistant cluster. So you can see this is pretty dramatically different. If we look to the lower cluster of resistant cells, that's dominated by the yellow clone, but also has significant amounts of pale pink and some other colors here, orangish. These clones are displaying a very different signature. They display ubiquitin-mediated proteolysis, changes in tRNA, biosynthesis and tRNA metabolic signatures. One of the questions we had was whether any of these changes that we detect in the post-treatment survivor cells were present in those clones pre-treatment. And so when we look back to that pre-treatment cluster, we kind of hoped that we would be able to see some of these signatures of which way the cells might go towards one or another resistant cell state. We hope that we could maybe see some of those indications in the pre-treatment state. And we really didn't find that in this middle pre-treatment cluster. There was no really convincing set of differences in these pathways. But if we looked at the cells that were 30 hours into the treatment already, there, because we know the future survivor cells and the future non-survivor cells, and when we compare those, which is shown in all of these dot plots, the non-survivors and the known non-survivors and the known survivors now, when we compare those subpopulations, we can see that there the future survivors are already showing some signs of distinct responses in these particular pathways that we had identified as upregulated in the post-treatment sample. So about 30 hours into the treatment, we start to see already the indications of which way those cells are moving and where they'll end up in these time points that are collected about 4 weeks later. There's some other interesting things to think about in terms of the way that these three subpopulations in the pre-treatment population interact with each other. And I won't talk about that series of studies, but we're just about ready to submit those in terms of, and this has been the work of Dee Gardner, another graduate student in the lab, who I think recently presented this at the Cancer Systems Junior Investigator meeting. I think some of you were also there, so I see some familiar names. But these three subpopulations seem to have some interactions in terms of their growth dynamics that we've been able to identify through recombination and sorting studies as well. So that's a sort of a new area that we'll be looking to move into and communicate soon. So to wrap up, I think these barcodes really expand the capabilities of current workflows that we're already using. They're compatible with cell lines, and I didn't show you, but we also have been using them in patient-derived cells. We have a project with getting patient-specific cells from Dell Medical School, which is here in Austin, looking at colorectal carcinomas. And so we've been able to use the same platform and workflows with patient-specific cells. The barcodes enable a study of clonal phenotypes, and importantly, those clonal phenotypes, but within the context of a heterogeneous population. So we're really starting to see how the cell behaviors change when they're in a heterogeneous population versus isolated in a homogeneous population, and the composition of the surrounding cell population seems to really matter for their phenotype and behavior. I'm excited that barcoding collections of cells really facilitates sharing and synergy across labs. So one thing we've been able to do with the barcoded 231 cells is send them off to the lab of Tanmay Lele at Texas A&M, which performs nuclear force measurements, or specialized technique that we really can't do in-house. So now we can compare the behaviors of many hundreds of clones in our own studies, and then in complementary studies with others, and do more with our from one population. I think we have some challenges with implementing these, and some opportunities, which are that the barcodes are, at least in my lab, starting to really inspire us to think about the structure in cell models, especially in vitro models, that we've kind of overlooked. maybe, and especially to think about how these models were initially derived, which, certainly sort of different cell lines may have started out as more or less diverse collections of cells, and we may be seeing indications of that now as we track their clonal dynamics. And I think for every cell library, we start to learn a little bit about how stable are its transcriptomes over expansion and over cell generations. It's a property that we start to call transcriptome diffusivity, just for the fun, I guess, of making up new words, but it has a fundamental biological interest, I think, and importance, but also a practical one, because we need to know that to know how long is a barcode a useful marker of transcriptome, right? So we're starting to be able to make those kind of measurements as well. So I think everyone might consider barcoding their cells, and please contact us if that's something that you'd like to use our tool to do, because we're happy to make that available and send you protocols and things to and help to get you started with that. My lab is really driven by graduate students, and certainly the projects that I talked about today were especially ones that were led by Aziz and then Kathy Gutierrez in the Wu lab, and Eric Brenner in my lab. And we're grateful for all of our incredible collaborators that work together so hard, long distance and through a pandemic to get this paper out, and happy to now stop and hear a few of your questions.
Okay, thank you, Amy. That was really great. We have so many questions. I will try to pick a few,
okay,
and then we send you the remaining ones so you could get back to the people
thank you
answering this. So there were repeated questions related to in vivo, like how would you, have you done any in vivo studies, and how would the in vivo and in vitro clonal, tracking and the drug sensitivity and what clones you select, would that be the same or not? I'm pretty sure it will be not, but
yeah, you and I talked about this. Yes, so we have done that with the breast cancer cells. We have implanted them and tried to see if they reflect the clonal dynamics that we saw in vitro. We've done it with the triple negative breast cancer cells. We've also done it with two different HER2 positive cell lines in a separate set of studies where we have in vitro data that I didn't get to show you today. And I can tell you that the cells that implant and give rise to the tumor in vivo are not necessarily the same ones that are surviving well in vitro. And so the pre-treatment conditions, even before we do drug treatment of the animals, were very different. And so this is the challenge, making these comparisons. Yes, that's a big challenge. We are doing some things with organoids and so on like this to see if that is a better model. But right now, that is the situation, is that this is the cells that survive and implant well and contribute to the tumor are not necessarily the fastest growing or most abundant clones in vitro.
Yeah, so then a couple of questions related to the drugs. Have you characterized the populations after multiple cycles of drug treatment?
Yeah, so in a separate set of studies, which I didn't talk about because we're not quite done analyzing, but we have done multiple cycles with our HER2 positive cells. Mm-hmm. We've done multiple cycles, and we have done multiple drugs in different order as well. And so I think that's going to be really interesting, but I'm just not quite ready to talk about the data yet.
Okay. And the question related to the plasticity of the high and low survivor populations that you were showing at the beginning with the leukemia, are these stable populations, or they can change into one another?
They are stable populations.
Mm-hmm.
They're stable. Mm-hmm.
Yeah. And I have a question on that, because I looked up the gene list that you presented that differentiated the high and low survivors, and you mentioned apoptosis related genes, but several of those genes like core-reticulin and some of the, the proteasome genes are also play a role in antigen presentation. Have
Okay.
you looked into that? Is there any potentially some immunological basis of the survivorship?
We haven't, but I have a different signature that comes out in one of the breast cancer data sets that's also related to antigen presentation. And, it's one of those that I kind of wondered if it was like a, weird spurious hit or if it was real. But if you're saying it's in both, maybe I need to look more closely at that one. That's really interesting.
Yeah, and that could be another reason why in vitro and in vivo could be different.
Absolutely, yeah.
Especially in patients. Okay, there are some very technical questions that, I can ask some like, when you do the infection with a library, do you use a particular MOI, and then do you need some passages before treatment? I think.
Yeah, so, and we have actually a chapter in methods of molecular biology now that's coming out. I just did the proofs, so it should be out soon, which details a lot of the technical points, but they're also in the first paper, the ACS paper in 2018. The MOI has to be low in the initial labeling of the cells. And this is actually an important point. We keep the MOI less than 0.1 so that very few cells are labeled then, right? But that also reduces the chance that you have 2 barcodes or 3 in one cell. And then we have a marker, a fluorescent marker, where we can collect the positively transduced cells. So the starting population for all the studies should be entirely barcoded cells collected based on facts using fluorescent marker. We didn't want to have any drug selectable marker to introduce more confounding factors with the cancer cells. But that does mean then you need to grow them a little bit. So you have, more than one instance of each labeled cell, right? Because you can't do much if you only have, 2 cells belonging to each clone. It's not enough to do any comparisons, and so you do need to expand. So that's right. Yeah, a lot of technical things to think about in terms of the
another question. Maybe we can have one more question related to the in vivo use and the neoantigens. Do you think the barcode itself would somehow generate immune response because of presentation? But I don't think the barcodes are they expressed. So could you get antigens from the barcodes?
Yeah, they're expressed as guide RNA sequences. I have I have not heard that. I talked to some I don't think so, but good question.
Well, the fluorescent markers themselves would be antigenic, so
that would be probably more of a worry, I would say, because that's much bigger piece, right?
Yeah, VFP, GFP are all very immunogenic, so that would be bigger issue than the barcode.
I think so. I agree.
Yeah. Okay, I think we have a number of other questions. One last one, because that probably multiple people interested in. What is the relationship between clone and transcriptional profile? Because it looked like on a UMAP plot that the same identical barcodes were not always identical transcriptionally.
Right. So I in the CL, so do you mean there's a little bit of cloud, right? Like they don't necessarily make a pattern together in the, So that's a great question, right? How big is the cloud that defines a transcriptional state, like, right? That's relevant for the behavior we're interested in. Here, it's drug sensitivity, and it seems like, at least in the CLL, we see, two particular clusters, and within that, the lineage supervised learning that Eric did starts to point to the fact that there's some subcluster structure that can be assigned to those lineages. But you're right, they're not overlapping. But it seems that, at least in that study with the CLL, we could assign well enough an unknown cell to a particular lineage based on its transcriptional state.
Yeah, and that way you could also use what is the epigenetic memory and what is not, that you could see like what is preserved, what is not preserved, and potentially identify regulators of that, if you could be tracking.
Yeah, that's right.
But
yeah.
anyway, all right, so I'm sorry, we have a number of questions that we will send you because we ran out of time to answer all of them.
Thank you.
Thank you again for this really great talk, and thank you everyone for attending the seminar and also for the very, active participation by the many questions and so on. So thank you. Bye.
Thank you all. Bye.